Hi everyone, thanks for coming today. So as Ali mentioned, I'm going to be talking about precision and personalized medicine. For those of you that don't know me, if any, I'm Shannon. I'm one of the PGY2 oncology residents. And thank you for the help of my content expert, Mike Fallon, for this presentation. So the objectives for this presentation are to be able to explain basic genetic and pharmacogenomic concepts and definitions, to be able to summarize the pharmacogenomic resources available to healthcare providers, and to be able to utilize pharmacogenomic resources in practice to enhance patient care. So to start off, I want to talk about the project that started it all, the Human Genome Project. So this started in October of 1990, also known as the ENCODE Project, and the goal essentially was to read the blueprints of human life and essentially to find out if all of the parts of the body do something functional. So it was an international team of researchers that sequenced and mapped all the genes that make up the human body. And this information has helped develop new ways to treat, cure, and prevent thousands of diseases, which we will talk about throughout this presentation. So to start off, let's talk a little bit about the human genome. So it's such a big concept, so I have a couple of things on this slide to help make it a little bit more relatable for some of the large numbers that you'll see on the slide. So the human genome has about 20,000 genes that code for a protein, and there's about 20,000 species of bees, is a fun fact. There's about 3 billion base pairs. And if you uncoil each of your DNA strands and you place them end to end, you can travel to the moon and back about 150,000 times. So that is pretty long, that is fitting in the cells of our body. There are about 733 genes identified that drive cancer processes specifically. There are about 700 species of extinct dinosaurs, FYI. And then BRCA, which is a gene that has been associated with breast cancer, I'm sure many of you are familiar with it, has around 69,000 variants for just one gene. And that's around the amount of words that are in The Notebook, for those of you that are romance fans, and for those of you who are not, around the amount of words that are in Frankenstein. So these are some of the pharmacogenomic biomarkers that are in drug labeling, so FDA drug labeling. As you can see here, these numbers aren't super huge. However, you might see some specialties that you might not have expected to have pharmacogenomic associated biomarkers. I know anesthesiology, gynecology, those types of things were a little bit surprising to me. But as we start to learn more about pharmacogenomics, about the human body, about genetics, you're going to start seeing the number of drugs on these tables go up pretty significantly. Just to put it into perspective, there are about 20,000 prescription drugs available in the US. So while these numbers aren't too big right now, definitely trending in the upward direction. And I will point out too, so I am going to focus on oncology a lot in this presentation just because there has been a little bit more funding going into pharmacogenomics and targeted therapies in oncology, and we know a lot more about it right now. So 221 is definitely the biggest number that you see on this slide, but this is the direction that medicine is going in general. So just to introduce, what is precision and personalized medicine? So we're all familiar with genetics, the branch of biology dealing with hereditary and variation of organisms. And then pharmacogenomics essentially combines the science of how drugs work, so pharmacology, with the science of the human genome or genomics. So we're using information about a person's genetic makeup to choose drugs, and then the dose and essentially the safety profile that's going to work best for that particular person. So what is precision medicine then? So essentially it's an innovative approach to tailoring disease prevention and treatment, and it considers differences in genes, environments, and lifestyles. So the goal is to target the right treatments for the right patient at the right time. So as you see this graphic here, so we have a patient group. So traditionally, as we know in studies, we have a patient group, we give everybody the same drug at the same dose, and it's essentially trial and error. Some people are going to have a really good response, some may not respond as well, some may have more toxicities. Versus if we use a precision medicine approach with pharmacogenomics, you're going to have some patients that are started at what would be considered the normal or the baseline dose. Some patients, based on their genetic profile, might be started on a lower dose, some on a higher dose, and some they might decide to exclude from the study because they're just not going to be a good fit based on their genome. So how can genetics impact healthcare? So going off that previous slide study example, we can definitely reduce medication toxicity. So we know if someone is a poor metabolizer for a drug, for example, we can either not give them that drug, switch them to someone else, or we can give them a lower dose. So they might end up with less medication toxicities and thus a decrease in overall cost of healthcare as well. Improvement in therapeutic efficacy for the same reason, which can also decrease the cost of healthcare because we're not spending as long on the trial and error phase of the process of figuring out what drug is going to work best for a patient. Genetics can also help with advanced disease screening, a more accurate measure to determine optimal drug doses. And then it is facilitating more drug discovery and production of targeted therapies, which we do definitely see a lot, specifically in oncology. So I'm not going to spend too long on this slide, but I just wanted to show you that it's not just genomics that is making huge changes in healthcare. There's a lot of other omics, they call them multi-omics, where a lot of research is going, and that may impact disease states and how we can make more targeted therapies. So briefly, epigenomics is one of these. So epigenetic changes in the cell. This can affect a person's risk of disease and how it's passed from parents to children. So that's an area of study. There's transcriptomics. So essentially transcriptomics is used to learn how genes are turned on in different cell types. So that can help different diseases as well. Proteomics is another study, so structure, function, and proteins, and then metabolomics as well. So there's a bunch of different factors, so disease or environmental, that may affect how metabolites are made and used in the body. So that's another area of study as well that is impacting practice. So I just wanted to go to the basics for a minute. So what are the key concepts that we need to understand the basics of genomics and pharmacogenetics? So here's DNA replication. I'm not going to bore you with this because we all took, I'm sure, several courses of biology in pharmacy school, but I bring it up here because it is really important to understand the fundamentals of biology and how DNA replication works, because it's also important to understand that this is where errors can be made essentially, and we can end up with mutations that can cause diseases. So for example, specifically on this slide, so we know that DNA polymerase is that enzyme up top here that binds to the DNA strands, and it adds DNA bases in the 5' to 3' direction. DNA polymerase is not perfect, and it can make mistakes, and that can result in mutations, as well as many other steps of this process can have errors. What is a chromosome? So thread-like structures made of a protein and a single molecule of DNA that serve to carry genomic information from cell to cell. We have 22 pairs of chromosomes and 1 pair of sex chromosomes. Histones, have it on the slide here, protein that provides structural support for the chromosome. So each chromosome has a long molecule of DNA. We have to be able to cram that into the cell somehow. So the DNA wraps around the histones, which makes it a little bit more compact. So as you'll see on this slide here, I have a chromosome. This is something worth paying attention to. So the way we name many of our mutations, especially as I've seen in oncology, is by where the mutation is, which makes sense. So looking at this chromosome, if it's a chromosomal mutation, we have the short arm, which is the top part. Abbreviation is P. The next part of it is the region on the top of the arm. So we have regions 3, region 2, region 1. And then within those regions, there are numbering of bands. So just typical 1, 2, 3, 4 band numbering within that region as well. And then numbering on the bottom is similar. So the long arm is abbreviated Q. And then you have your 1 numbered region, and then you have bands within that particular region. There are a couple of different types of chromosomal mutations. We have deletions, so as you can see, just a segment is lost. Translocation, so a segment from the chromosome is transferred to another. Duplication, so a segment from one chromosome is transferred to its homologous chromosome, so it's giving a duplicate of some genes. And then we have inversion, so a segment of a chromosome is inverted. What is a gene? So going back pretty basic here, so genes are our basic unit of inheritance. As we all know, they're passed from parents to offspring. About 1% of the genome are proteins that code for a protein. So that's a pretty small portion of the genome. Looking at transcription and translation. So these are important steps, and this is again where mutations and polymorphisms can arise. So transcription occurs when a strand of DNA is used to build a template RNA. You can see that on the slide here. It starts at the promoter region. And then we have translation. So this is where mRNA is translated into amino acid sequence, which can then be used to build a protein. We'll talk a little bit about that a bit more in a couple of slides, but there's 3 steps, as I'm sure you all remember: initiation, elongation, and termination. So then we have mutations and polymorphisms. So both have some abnormality within the genome, but it's a little bit different. So we have mutations, which are rare permanent alterations of a nucleoside sequence of a gene that's caused by intrinsic or extrinsic damage. These typically try to fix themselves with proofreading mechanisms via replication and the repair process. So homologous, non-homologous repair. I'm sure those probably sound familiar from our biology days. And then we have polymorphisms. So these are a little bit more common. These are loci in the genome that have a varied sequence within a particular population. So the least common sequence is 1% of that entire population. So examples here, so mutations, we would have sickle cell anemia, Klinefelter syndrome, Turner syndrome, and then examples for polymorphisms, we would have human sex and APO blood group. Going back to the transcription translation type stuff that we talked about. So when a protein is being translated, there might be different mutations. So an amino acid may end up being switched for an amino acid, or something might happen that causes a change in the sequence, which can ultimately impact the protein. So some of the mutations that you'll probably see, so you have some nucleotide sequence change, you have an insertion or deletion, loss of gain of chromosomes, something happens, and that can lead to missense mutations. So one base is changed and that results in a different amino acid. You can have nonsense mutations, which one base is changed and that results in a stop codon. That would cause truncation of the protein and oftentimes a non-functional protein, so loss of function or protein with minimal function. Can also have a frame shift. So if one base is added or deleted, instead of reading that 3 pair that it typically reads, it's going to shift the frame over and it's going to read a different pair of 3. So that can result in an altered amino acid sequence. There's also gain of function and loss of function mutation. So gain of function produces a novel function or increased protein product, and loss of function reduces the level of activity. So we'll kind of come back to this topic, but essentially, especially we see in oncology, it's a lot easier to treat a gain of function because there is something to actually treat, versus if someone has a loss of function mutation, it's a lot harder to target. So moving into testing for chromosomal and gene mutations. So some of our testing for chromosomal mutations are spectral karyotyping, or SKY. And then what I see more commonly in oncology, at least, is fluorescence in situ hybridization, also known as FISH. For gene mutations, we have next generation sequencing, which we'll talk a bit about within the next couple of slides, polymerase chain reactions, restriction enzymes, and microarrays. So this is just an example of FISH analysis. So this is what it looks like when we get the results back here. So this is for a patient with acute myeloblastic leukemia, and we send these out. This FISH analysis will help kind of guide us to whether they're low risk patient, high risk patient, that type of thing, but this is what it would look like. There are other types of mutations as well. So we have somatic versus germline mutations. Germline mutations are changes to your DNA that you inherit from the egg and the sperm. So as you can see here, so these are our gametes. And then we have somatic mutations. So these are changes to the DNA that happen after contraception to cells other than the egg and the sperm cells. So these are essentially everything else. They're diploid and they contain about 46 chromosomes for the somatic cells. And then I just wanted to briefly touch on CYPs, so cytochrome P450 superfamily. Why do we care? So obviously in our day-to-day life as pharmacists, we see drug interactions, but as genetics and pharmacogenomics is starting to become more of a thing, we need to consider what type of metabolizer a patient might be for specific enzymes. So this table up here is just a general, on average, CYP2D6 prevalence for metabolizer polymorphisms, but on average, about 5 to 10% of patients are poor metabolizers, 20 to 40% of patients are intermediate, 60 to 80 are extensive or normal, and then about 1 to 5% are ultra rapid metabolizers. So what does that mean? Why do I care about that? So let's say you have a patient who is going to be taking venetoclax. That's an oncology drug, and it's metabolized by CYP3A4. Usually when patients are on venetoclax, we also give them posaconazole. So as an azole, that's a CYP3A4 inhibitor. If we then have the genome for this patient and we know that they're a poor metabolizer, what do we do with that information knowing that even if they didn't have that poor metabolizer, we typically reduce the dose of the venetoclax with the posaconazole. So it can be really complicated. And definitely something great as pharmacists, we can be on the front line of this and understanding how to best guide therapies, especially when we start to know a little bit more about the genome and we start to get that information on patients up front. This is a pie chart I thought was really interesting. So this is the number of polymorphisms associated with each CYP. So for example, again, looking at CYP2D6, there are 114 different polymorphisms or 114 potentially different ways a patient may respond to different medications based on the way their particular CYP2D6 will metabolize medications. And then I wanted to touch briefly on star alleles because this is something that you'll see, especially if you're looking at some of the genomics guidelines. So say we're talking about CYP2D6 again. So CYP2D6 star 1 would denote the default reference haplotype. Just to remind you, haplotype is a set of DNA variations or polymorphisms, and it can kind of refer to a combination of alleles or a set of SNPs on the same chromosome. But say, so star 1, that's normal, that's wild type. All the other star alleles will denote different haplotypes where different variants have been observed. So a star 2 or a star 3 might denote a loss of function, a gain of function, some other different type of variant. As you can see here on the examples of 2D6 diplotypes, a lot of the guidelines will show you, and this is an activity score, so relates to the phenotype classification system. If a patient has, say, star 4 and star 10, they're an intermediate metabolizer. Or if a patient has star 3 and star 4, they might be poor metabolizer, and that might be actionable depending on what type of drug they're given. So that's just something to keep in mind and to know your references for. The particular number, so like star 2 or star 3, doesn't associate with whether or not someone's a poor or intermediate metabolizer. It's the order in which the particular variants were discovered. So definitely translation tables and guidelines like this are definitely your friend when you know the type of diplotype that the patient has and you're trying to figure out what type of metabolizer they are. So moving into my first question, so we talked about naming some of the mutations a little bit. So one of the diagnostic mutations in acute myeloid leukemia is this lovely slur of numbers and letters right here. What does that mean given these 4 options? All right, so I'm seeing an A in the chat, which is right. So it is an inversion of chromosome 16. So INV is inversion, chromosome 16, between the short arms, that was what P was abbreviated, region 1 band 3, and long arm 2 band 2. The other ones won't be that tricky, I promise. So moving on into the next portion. So next I want to talk about next generation sequencing. So next generation sequencing is called massively parallel sequencing. So essentially offers ultra high output scalability and speed. We used to use something called Sanger sequencing, which took a little bit longer, was a little bit less accurate, and you needed much higher sample input requirements to be able to detect the mutations. Essentially, they look at hundreds to almost thousands of genes at one time in a bunch of different samples, and then they can analyze different genomic features in a single sequencing run. So this has been extremely helpful, especially in oncology. So I just want to go through a couple of the NGS panels that we use in oncology. I have one for other specialties as well, and I'm sure we'll be seeing more for other specialties as this becomes more commonplace. So this first one is Strata NGS. So it's a 429 pan-solid tumor assay. They market themselves as being able to use about 1/10 of the tumor tissue typically required for sequencing tests. And they co-isolate both DNA and RNA from the same tumor sample. So looking here, so they'll give you the genomic findings, and then if there's any associated therapies that you can use. So this particular patient had a ROS1 fusion mutation, and so ceritinib, crizotinib, and entrectinib are all therapy options. And as you'll see on the bottom here, there's some other biomarker findings, and these would be associated with therapies on the right-hand side here if the patient did have either of those mutations. Pretty similar information, but it just kind of tells you what qualifies them, where says it's okay to use these drugs. So especially for insurance coverage, it's helpful when the FDA says this is a legit indication. But it'll also show you if there's active clinical trials for any of the medications, and then NCCN is the oncology guidelines, so that's good as well if they recommend it. And then on the bottom, you'll see here, so there's more biomarker findings. So this patient had PD-L1 high, which is just another marker, and then they have more associated therapies that they can use because of that. Here's just a quick snip of all of the mutations that Strata tests for. So Strata is very, very helpful in solid tumors, and it does test for a lot of different mutations. Next is a Guardant 360. So this is a liquid biopsy laboratory test. So essentially they take a blood sample and then they look at the DNA that's found in the circulating cell-free DNA. So pretty similar information, just a little bit of a different layout. So it'll give you the mutation that the patient has, the biomarker. So this particular patient had an EGFR mutation, so an exon 19 deletion. It'll show you the associated FDA-approved therapies and if there's any clinical trials available. Again, it'll show you some of these other biomarkers down at the bottom here, and then it'll also give you a tumor response map, and it'll show you how much of that particular mutation was amplified in the DNA. It also gives you this type of table, which is really helpful. So if there's any clinical trials open for that particular mutation that the patient has, it'll give you the trial ID, the title, and then the sites in which the trial is being conducted. Here we also have a myeloid malignancy panel, so next generation sequencing assay, so it screens for DNA alterations and then RNA fusions, and it looks at about 50 genes. So as you can see on here, so these are detected alterations of potential pathogenicity. So this first one, TP53, that probably sounds familiar to most of you. I feel like that's the one thing they teach you in school that can cause cancer. It's a tumor suppressor gene, so when it stops working, then the tumor is not being suppressed. It'll show you the type of alteration, so it's a nonsense mutation, which we talked about. A stop codon being placed a little bit earlier, so the protein is truncated and oftentimes it's loss of function because the protein isn't fully formed. It'll show you that's pathogenic, it'll give you therapeutic implications, and then it'll give you some other information as well. These are all of the genes that the myeloid panel tests for, which is also quite a few. Next, we have this Oncotype DX. So this is a recurrence score for hormone receptor positive breast cancer using RNA that's extracted from the tumor tissue. So essentially it analyzes the activity of about 21 different gene types that can influence how likely the cancer is to grow and respond to treatment. And we can use that information to determine whether or not the patient is just going to stay on hormone therapy or if they would need the addition of chemotherapy. So as you can see here, it'll give you the recurrence score, the likelihood of recurrence at 9 years. And then it'll give you some other information as well, which we'll talk about in a little bit. Here is the 1 ohm. So this isn't something that we use in oncology, and I included this because I know some other sites do use it for their non-oncology specialties. I know specifically the Mayo Clinic will use this. They have a pharmacogenomics clinic where pharmacists are consult, and they rely heavily on this type of panel. So it's a clinical laboratory test. It's out of Minneapolis, Minnesota. It's a cheek swab, and you can essentially order custom panels or single SNPs. So you could create like a custom psych panel or a custom cardiac panel, or if you just wanted to look for something specific, so you only care about warfarin, you just want to see the B core C1, you can do that as well. So this is really helpful. Okay, so moving into resources. So what tools are available for pharmacists to help understand and make pharmacogenomics recommendations? So first I want to start out with some of the resources for non-oncology pharmacists. So first off, we have the US FDA, so our package inserts. I'll show you some of the information that's available from the FDA, but they also have biomarker and association tables, which we'll talk about as well. CPIC guidelines, we'll talk about a little bit. Dutch Pharmacogenomics Working Group guidelines. This is just another big group where they'll publish guidelines on if a patient has a specific mutation, how does that impact drug therapy? So those are really great references. The American College of Medical Genetics and Genomics is a good resource. So it's a team that works on developing guidelines, laboratory services, databases, population screening guidelines, and then just making more uniform laboratory standards. PharmGKB we'll talk a little bit about. That's a really great resource. There's PharmVar Consortium and ClinGen, and these are a little bit more of the science and less of the clinical interpretation of pharmacogenomics, but they do have some of that information as well. So starting with our first resource here, so these are the CPIC guidelines, also called Clinical Pharmacogenomics Implementation Consortium, designed to help clinicians understand how available genetic test results should be used to optimize drug therapy. And essentially their tagline is they're prepping for the future where preemptive genotyping is common and healthcare providers will always have genetic information on hand. That's kind of partially what I'm hoping to do with this presentation too, is just show, I know a lot of it is oncology focused just because oncology does deal with a lot of pharmacogenomics on the day-to-day and not all specialties do, but eventually, hopefully, preemptive genotyping will be the norm. So you will see all of these resources in some type of form in whatever specialty you practice in. So when you go to PharmGKB, this is what the website looks like. It'll give you information. So it has a prescribing information section. So it'll give you, if you look for a specific drug or a specific mutation, it'll give you associated CPIC guidelines, Dutch Pharmacogenomics Working Group guidelines. It'll give you, if there's any FDA label annotations, it'll have those cited as well. It has pathways, it has clinical and variant annotations and literature. So if you're looking for the primary lit, where is this recommendation actually coming from, it has that as well. And then it also has something interesting. It's called very important pharmacogene summaries. So they're essentially gene summaries for non-experts, which can be really helpful if you're learning about pharmacogenomics or for patients. Then they have this little pyramid. So on the bottom, they have the primary pharmacogenomic literature, and on the top, they have all of the guidelines and how to implement it. So PharmGKB does a really good job, and they try to cover all of this starting from the bottom. Where is this information coming from to, okay, now that I have all of this information, what do I do with it? So this is an excellent resource. Next, we have the FDA association table. So essentially, the FDA has looked at all of these drugs and all of these genes, and they've decided that there's enough evidence to suggest that patients with certain genetic variants of these genes might have altered drug metabolism or just some type of different therapeutic effect. So that is worth noting if you do have the pharmacogenomic information. It's not saying that all of these are actionable and that you have to test for all of these, but there is sufficient evidence that there is some link. So if you do have that information, it might be worth considering. And then we have the FDA biomarker table. So it's a very similar table, and this is what we talked a little bit about in the beginning with like even anesthesiology had some notable biomarkers. But this table is essentially drugs that had some type of biomarker mentioned in the package insert. So again, while not all of them are necessarily actionable, it might be worth, if you do have a patient's pharmacogenomic information, just kind of taking a look at this and just seeing if there are any associations. And then you can look at guidelines and see if there's anything that you should be doing about that, if any changes to therapy are needed. Next, I have ClinGen. So this is a great resource if you're looking for germline or somatic mutations. So we kind of talked a little bit about it, but there's a couple of different types of germline mutations. So the two kind of main ones that I think of for germline mutations are sickle cell and cystic fibrosis. So let's look up cystic fibrosis. So this is the transmembrane protein that is mutated in our CF patients. So it'll pull up this slide here. So it gives you a little bit about the gene summary. It'll give you a haplotype insufficiency score if there is information on that. And then it'll give you this very specific genomic view. So this is a little bit more useful if you're doing research versus clinical, but if you're trying to design a primer sequence or look very specifically at like the base pairs or the amino acids that code for the mutation, this lets you zoom in really far into the gene and you can learn a lot from looking at it. So those are kind of the main resources now for non-oncology pharmacists. And now I'm going to cover the resources for oncology pharmacists. Again, while this information may not be pertinent today for all of you, as preemptive genotyping hopefully does become more common. I know, like for example, St. Jude preemptively genotypes all of their patients and they have that fully integrated into their EHR. So hopefully that is the future of pharmacogenomics, but you might see more resources similar to what we have in oncology in your particular specialties. So resources that we use in oncology, so again, our NCCN guidelines. So these are our HEMONC treatment guidelines, but they do have pharmacogenomic recommendations implemented into them. So that's a great resource for us to look at. We'll spend a couple of slides looking at OncoKB, My Cancer Genome, and CBioPortal, ClinVar, Nomad, and then BRCA Exchange as well. So this first resource that I'm going to show you is OncoKB. So it's a precision oncology knowledge base. It's powered by Memorial Sloan Kettering, which is a huge cancer center in New York. They have over 5,000 cancer gene alterations noted on this particular website. And this is also used at Tumor Board. If, for those of you that aren't familiar, that's where providers can present particularly difficult patient cases where other providers, pharmacists, can weigh in on those cases, and oftentimes, especially within oncology, because there's so much genomics are brought up, and OncoKB can be used. So to learn how to best use this resource, I have a little case. So TK is a 70-year-old male who has been taking afatinib for metastatic non-small cell lung cancer. Does not matter if you've never heard of that drug before. His oncologist has determined that his disease progressed, and he ordered a new strata report to see if he has any new actionable mutations, because tumors can change and make new mutations. Report comes back positive for T790 EGFR mutation. What do? So you can go to OncoKB. You can look up this particular mutation. If you're wondering what is this mutation, and then it'll pull you to this screen. So there's a couple of levels of evidence. So there's level 1, which is green. You can use this drug. This is great. Level R1 means that a drug is going to be resistant to the mutation. And then FDA level 2 is just the level of evidence the FDA has associated with this recommendation. So as you'll see here, we have green. So T790, osimertinib is an appropriate drug. And then as we can see, the patient was on afatinib, and it looks like T790 is a mutation. Patients that might be resistant if a patient is on afatinib. So using this information, you recommend to the oncologist that the patient should be on osimertinib now, and you let their oncologist know. Moving into our next resource. So this is My Cancer Genome. So it contains assertions on the impact of almost 17,000 molecular biomarkers and the use of almost 3,000 drugs in around 950 cancer types. So a lot of information is contained on this particular website. A lot of different things we can look at. So we'll start off with clicking clinical trials, and then it'll bring you to this slide here. So you can sort by recruiting status, phase, disease, biomarker, and drugs, and it'll give you all of this information about open, or if you click recruiting, depending on what you click, might be another phase in the trial, but trials that you can look at for that particular mutation. Then if you look at diseases, you can sort by disease hierarchy or biomarker, and it'll show you biomarkers associated with a particular disease. You can look at biomarkers, and you can sort by biomarker type, disease, and then drug category. And then within each specific biomarker, so you might get a report back with a biomarker that you've never seen before, and it didn't necessarily suggest a treatment. You could look in here to see if there's any clinical trials that maybe it didn't pull. And then it'll just give you a little bit more information about it as well. There are a couple other resources on that website, talks about drugs specifically and pathways as well, but those are kind of the main things that we use in practice. So this next resource is called cBioportal. So it's an open source software system, and it essentially provides an analysis of large scale cancer genomics data sets. So what does one do with that? So let's say we looked up an EGFR mutation. So the first page that comes up is this. So it'll show you the alteration frequency based on cancer type. So as you can see here, a patient with lung cancer is much more likely to have an EGFR mutation than a patient with thyroid cancer, for example. And then if you scroll a little bit further down, it'll show you this. So you can look at the lollipop plot. You haven't heard that term before. I think they're kind of fun looking, but it's by variant, so easy to see hotspots, exon mutations, mutation types, etc. So we kind of talked about some of those mutation types, and as you can see on the side, they have missense, truncating, in frame, some of those things that we kind of talked about. Next, we have ClinVar. So let's use another example. So let's look up BRCA2, and as we talked about in the beginning, BRCA is a mutation that can be associated with breast cancer. So again, it'll give you another lollipop type graph. So it'll show you if the mutation is pathogenic, if we don't really know, if it's benign. And it'll also give you more information on the bottom here if you know specifically what gene within BRCA2 that you're looking for. Nomad is another good resource, so also known as genome aggregation database. It's essentially like a cloud for pharmacogenomics, but it was developed to aggregate and harmonize both exome and genome sequencing from a wide variety of large-scale sequencing projects. Essentially, one of the more useful components to this particular resource is that we can assess the chance that a particular mutation is currently known to be germline related. So let's say we look up TP53, which is that tumor suppressor gene. We're going to come up here, so it's going to give us our ClinVar variants, it's going to give us our Nomad variants. So let's go down here to the bottom, and we're going to pick a variant ID. So let's look at this VAL31IE mutation. So we'll click that, and that'll bring you to this page here. So it'll give you the age distribution, and it'll give you the population frequency for that particular allele. So if you're wondering, so say the patient is of European descent, and you're wondering if that particular mutation is common within that population, you might want to look here. You could look at the allele frequency, and that might help to guide whether or not it would be recommended for the patient's family to get tested for that particular mutation. And then next we have BRCA Exchange, which we talked a little bit about. So basically aims to advance the understanding of the BRCA mutations. It's not only in breast cancer, but also ovarian, pancreatic, and a couple of other cancers as well. So let's say we're going to look up BRCA1, and that'll bring you to this slide here. So it'll show you if the mutation is pathogenic, of little clinical significance, or just uncertain significance. So it'll bring us to our second assessment question. So HL is a 64-year-old female who is starting treatment with Lumakras, an oncology medication. Based on FDA indication, it can only be used for patients with a specific tumor type for metastatic lung cancer. Which of the following resources could you use to learn more about this particular mutation? All right, I'm seeing E in the chat, which is great. It is all of the above. So just to wrap up, I wanted to go through some of the resources that we talked about using clinical scenarios. So first wanted to touch on some of the other specialties because I know we spent a decent amount of time talking about oncology and not everyone is an oncology pharmacist here. So I wanted to touch on some other subjects. So on this slide, I wanted to focus on voriconazole. So CYP2C19 is the main pathway of metabolizing for voriconazole. So up on the top of the slide here, this is from the CPIC guidelines. So again, it just is kind of the average percentage of the type of metabolizer for that particular enzyme, and then the examples of the diplotypes associated. And these are some of the recommendations from CPIC, so implications, recommendation, and strength. It's also worth noting that the Dutch Pharmacogenomics Working Group has recommendations for CYP19 mutations in voriconazole as well, as does the Infectious Disease Society of America, which is really interesting, seeing such a big name guideline have pharmacogenomic recommendations. And essentially what the IDSA said was that some polymorphisms of CYP2C19 might result in different therapeutic outcomes and safety profiles for patients that are on voriconazole. So if you do have pharmacogenomic information, then you should be using it for these patients. Just taking a minute to look at some of the primary literature. So there's a lot of studies on this. This first one looked at patients, so CYP2C19 guided voriconazole prophylaxis in patients with neutropenic acute myeloid leukemia. And so they dosed based on the type of metabolizer the patient was. And essentially what they found is that doing this type of dose guiding is feasible, and it could result in reducing the risk of subtherapeutic trough concentrations that can cause breakthrough fungal infections for patients on voriconazole. This was a very similar study looking at patients getting a transplant, and they essentially found similar results. And they also looked at cost in this study and believed that the cost of getting the genome type testing was better than the cost of breakthrough fungal infections due to patients with subtherapeutic drug concentrations of the voriconazole. Next, we have depression. So as we know, it can be really challenging to get patients on the right antidepressant medication. They take about 6 to 8 weeks to work for a patient, and then if you wait 6 to 8 weeks and that one wasn't for you, you have to start over, and that's another 6 to 8 weeks before you feel better. So that can be a really long road for these patients. So if we are able to get preemptive genotyping for these patients, we might be able to mitigate some of that trial and error period. There's a lot of great guidelines, especially for the SSRIs, so CPIC guidelines and Dutch Pharmacogenomic Working Group have great guidelines as well. This is an example of the CPIC guidelines for CYP2C19s, again, specifically just because a lot of the SSRIs are metabolized through it, but they have great recommendations for these patients as well. Warfarin also has pharmacogenomic implications, so CYP2C9, VCOR, CYP2C, and CYP4F2 are all linked to either increases or decreases in warfarin sensitivity. So the first 3 mutations on this slide would result in needing a lower dose, and then this bottom one results in needing a higher dose. Something interesting too, it's not just warfarin within cardiology. There are actually a lot of medications that they're starting to find have pharmacogenomics implications. So for example, the Mayo Clinic has something they're calling a focused pharmacogenomics panel. So that includes many CYPs, it includes VCOR, and then it also includes SLC01B1, which is the enzyme that metabolizes statins. The FDA has recommended guidance for warfarin dosing based on genotype if known. I wouldn't have thought they would have updated this package insert because warfarin is so old, but they did, and there are actual recommendations. So that's a good place to look if you have that information. And there's also a ton of different warfarin dosing calculators that you can use if you know a patient's pharmacogenomic information. And then here's just another example using oncology medications. So mutations can be resistant to drugs, as we know. So here we have, many of you may have heard of the Philadelphia chromosome in school. BCR-ABL is the abbreviation, but it's a translocation between chromosome 9 and chromosome 22. Tumors are super smart and can develop resistance mechanisms. So as you can see on this very colorful graph chart here, it'll show you whether or not these different agents which can treat this mutation might be sensitive or resistant to the medication. So based off information like this, our NCCN guidelines have treatment options based on mutation profiles. So whatever specific mutation the patient has, you would use a different drug that would treat that mutation for the BCR-ABL mutation as a whole. So kind of wrapping up here, so this is our final question. I wanted to give you a case where you had all of the information you needed, but just to be able to apply pharmacogenomics concepts. So JT is a 38-year-old premenopausal female who was recently diagnosed with hormone receptor positive breast cancer. Her oncologist asks you what you would recommend for therapy based on her oncotype score. What would you recommend? And I'll give you a couple of seconds to look at these charts. All right, and I'm seeing B. Great. So we're coming to the end of this presentation. Just to wrap up really quickly, there are obviously a lot of different barriers for pharmacogenomics. So evidence barriers, logistics barriers, financial barriers, knowledge barriers, ethical barriers. Something we'll talk about on the next slide extremely quickly is provider and pharmacist comfort level. Training infrastructure, it can be operationally challenging. The turnaround time for in-house versus send-out tests can be challenging. There's different guidelines on when to do testing. Cancers and other disease states evolve, so how many times can you get insurance to pay for tests on these patients? And then there's about 20 advanced NGS platforms. The hospital's not going to use all 20. What are we going to use? Does it have all the mutations that we want covered? So it can be really, really complicated working through these barriers. There have been a couple of studies looking at barriers identified by healthcare providers. So this was a study that was done by our lovely oncology pharmacy team, looking at comfort level and assessing pharmacogenomic data's impact. So we'll leave this here for a second just so you can glance over it. But looking at understanding next generation sequencing panels, cytogenetics, genetic driven biomarkers, and then being able to use that information, looking at turnaround time, insurance denials, things like that. So we can get really complicated. And then there was another study looking at physicians, and essentially what they found was that about 3/4 of respondents didn't get any pharmacogenomics training in school, and then about 90% of them didn't get any postgraduate credits in pharmacogenomics. So how can we expect healthcare providers who have never been given this information to be able to effectively utilize it in practice? So to summarize, precision and personalized medicine is a massively growing field. Oncology is particularly advanced, but it is a good model for what it's going to look like in the future for other disease states when we start to do more preemptive genotyping as a whole. Tons of different resources available, and pharmacists are uniquely trained to be on the front line of this change in practice. Definitely not a ton of education, definitely more now going on in both pharmaceutical and medical education, but we're definitely uniquely situated here to really help out when this starts to take off. So these are my resources. Thank you so much for listening. I'll just leave this slide up here, but what questions do you have for me?